on Intruder Alert, the innate immune response, organized by the Kerala Biotechnology Commission under the Kerala State Council for Science, Technology and Environment. As you know, Kerala Biotechnology Commission is the agency under the government of Kerala to promote biotechnology-related activities in the state. And this session is part of KBC's ongoing efforts to promote scientific awareness and interactions and dialogues in frontier areas of biotechnology. We are truly delighted to have participants from academia, research and industry joining us online today. And to begin the program, I take this opportunity to extend a warm welcome to our distinguished speaker, Dr. Manoj V.M., who's a scientist at College of Medicine, University of Kentucky, USA, who has kindly agreed to deliver today's expert talk. And I also take this opportunity to extend a warm welcome to our Member Secretary, Professor A. Sabu, who has been a guiding force behind KBC's all efforts. A warm welcome to all the participants who have joined us from different institutions and organizations. And to begin with, may I now invite Professor A. Sabu, the Member Secretary of KCST, for the opening remarks.
Good afternoon, everyone. Very good morning, Dr. V.M. Manoj, scientist from the College of Medicine, University of Kentucky, USA. First of all, thank you, Dr. Manoj, for accepting our invitation to give webinar. I know that it is all time for you to be online for our interaction with the interested people from the state of Kerala. In fact, the Biotech Commission attached to Kerala State Council for Science, Technology and Environment is having several initiatives to bring in the Kerala diaspora from across the globe, either online or offline. And Dr. Sarika and her team is successful in that. And Dr. Manoj's lecture today is part of that effort to bring in more people from Kerala to the activities of Kerala Biotechnology Commission. I will not take much time. Let me tell you something about the Government of India launched the Bio E3 program, the biotechnology for economy, employment and environment. Attached to that, all over the country, the states are taking up this activity, adopting the Bio E3 cell policy of Government of India and starting Bio E3 cells in different states. So totally Kerala also will be taking it up and adopting Bio E3 cell policy, so that there will be a better alternative by the green way of synthesis manufacturing and all through bio-based enterprises. So Kerala, with its rich natural resources and well-trained HR, will be able to come up in a big way as far as biology-based economy or biotech-based economy. And of course, we need guidance and support from the experts working across the globe of Kerala origin. I hope Dr. Manoj also will be able to help us, guide us in future, also to go ahead with the activities as far as biotech development is concerned in Kerala. So thank you very much, Dr. Manoj, once again, and thank all the participants for their interest in listening to Dr. Manoj. You will be speaking on innate immune response, the signaling pathways and all. I understand that Dr. Manoj is basically a chemist switched over to the biological science area. So once again, thank you very much, Dr. Manoj, and thanks all the participants. Thank you.
Thank you so much, sir. Now, my pleasure to introduce our distinguished speaker for today's session, Dr. Manoj V.M., the scientist at College of Medicine, University of Kentucky, USA. Dr. Manoj has extensive experience in molecular immunology and innate immune signaling, with more than 30 publications in reputed journals. His pioneering work on toll-like receptors and instinct pathways has provided key insights into antiviral defense and inflammation. Today, he'll speak on the topic Intruder Alert, the innate immune response, And this is a particularly timely topic as the 2025 Nobel Prize in Physiology or Medicine is also recognized the discoveries related to immune tolerance, underscoring the global importance of understanding the immune regulation. Sir, we are honored to have with us today. The screen is yours. Please, sir.
Okay. Can you hear me?
Yes.
Yeah, first of all, thank you very much for the introduction and also inviting me to offer this talk. Yeah, so the topic is about the innate immune response. So before going to the talk, I will. Yeah. Yeah, so this is the outline of today's talk. So I will give the introduction touching this innate immunity, and then we'll go to the and we'll touch my research area and then give the future direction. So we heard the word immunity recently, mainly recently because of this COVID-19 and also related to the Nipah. And also we heard some words like antibodies, HCQ, vaccines, cytokine storm, and also we are talking about this long COVID. And recently we heard this word like brain-eating amoeba. So what is immunity? So before going to the immunity, so there are some keywords for this immunity. So the first one is about the innate immunity. So the innate immunity is the first line of defense against any intruding pathogens. So we can say that, for example, it's the skin, mucous membranes. They are the initial barriers. And after that, then it's coming to the cell level. So, and then from there, it's going to the deep, other cells are recruiting for the other one. So the second term is called adaptive immunity. And this one is, so after the infection, then our body is fighting against these intruders. And also we can acquire this defense through the vaccination. So these terms are, this is called adaptive immunity. Adaptive immunity, usually it's last for a person's entire life. Like most of the vaccines we are taking in the childhood can protect us from through our entire life. Then another term is called passive immunity. So in the passive immunity, we heard that recently, like during the rabies or the dog bite, or in the Nipah case, we are giving antibodies to the patients. So this is like, so usually this innate immunity and adaptive immunity to fight against a microorganism will take time or days. So, but in conditions like in the Nipah or in the dog bite, we need to rescue the patient immediately. So we are giving the antibody, so antibodies to the patients. So these antibodies will also protect against this microorganism, but it will last only for a few weeks or months. So these are the keywords mainly coming into this immunity. So in the innate and adaptive immunity, there are, as I mentioned, there are so many immune cells are involved in this innate immunity and adaptive immunity. So mainly in the innate immunity, there are macrophages, like we heard about, we know about the white blood cells, neutrophils, basophils, dendritic cells. So these innate immune cells will recruit these other adaptive cells, like B cells or T cells. B cells will produce antibodies, and T cells will. Recently, we heard about these T cells as said. This Nobel Prize went to this the work related to the T cells. So it's regulatory T cells. So they can also in the regulatory T cells, they are protective in the autoimmune diseases. So I'll come to the it's a later part. So these immune cells. So what is happening in there? So these actually these pathogens, they are having the proteins and nuclear parts in these pathogens. So our body or in the living organisms, the body is or the defense system is targeting these proteins or these nucleic acid particles. So they are called PANs. And then, so for these PANs, we need a receptor on these immune cells. Once this receptor recognizes these PANs, then it will start signaling, and then it will releases cytokines and other proteins. So this is the structure for the innate immunity signaling. So this is a broad picture of what is happening when this microorganisms are attacking our body. So there are these receptors. This is the cell wall, so a plasma membrane. So there are receptors on the plasma membrane which can recognize different parts of this microorganisms. And also there are receptors inside the cytoplasm. So in the cytoplasm, there are sensors for DNA and sensors for RNAs. And also there are these receptors inside the endosome. So what they will do once they recognizes this PANs, then they will get activated, and then will send the signal, so recruits the proteins downstream and go to the downstream signaling, and will end up with mainly three pathways. One is interferon pathway. The second one is the NF-kappa B pathway and the MAP kinase pathway. So these pathways will lead to the pro-inflammatory cytokines or type 1 interferons. So once in the, so this is the condensed picture of what is happening when a virus is going inside. So I am mainly targeting the cytosolic part. So there, the DNAs can detected by the sting, or the RNAs by the regale-like receptors, or the nucleic acid particles like DNA or RNA inside the endosome. So as I mentioned, when the main downstream of this one is interferon and NF-kappa B pathway, which leads to the antiviral properties or the pro-inflammatory cytokines. So if we are looking at the interferon, so once the interferon is produced, then again it will release into the outside the cell. So this interferon can so the interferon, the cells are having this receptors to detect this interferon. So once the receptor finds this interferon, then again it will activate the downstream signaling, and then will release or induce some proteins called ISGs, interferon stimulated genes, and they are actually the antiviral proteins. So once ISGs are produced, then they can restrict the viral replication. But again, this interferon and NF-kappa Bs are like a double-edged sword. So it's having a positive and a negative roles in this interferon path. I mean the signaling pathways. So coming to the so how this virus can enter into this cells. So they need an anchor. So we heard about that this COVID related thing. So the COVID is having the spike protein. So, but this spike protein, they need to get an anchor on the cells through the ACE2. So we are giving the vaccine actually to block this. So they are targeting the ACE2 so that this virus cannot attach to the ACE2. So once it's attached, it can go inside the cell through the endosome, or it can inject their nuclear particles into the cytoplasm. So after that, then they can propagate by hijacking the host cell functions. So this is the detail of that one. I am not going through this, but it's actually going through the transcription and the translation, and then will produce more proteins, and then more that leads to more virus, and then it releases. So once the like, for example, RNA is viral RNA is detected by the receptors, so then they can, as I mentioned, they can go through the interferon ISGs, or it can also kill the cells. So that's why I initially said that about it's a double-edged sword, because the cell death is also a good thing. It can remove the virus, but if the cell death is more, then that will lead to the disease, like that's happening to the like respiratory virus. If the virus is more, then that leads to the lung damage, and that leads to this death of the host. So the second pathway is the NF-kappa B. So NF-kappa B actually binds to the DNA and then transits this inflammatory gene expressions. Again, the inflammation is required for the host defense, but the same thing, if it's high, it's out of control, then that will harm the host. And these viruses are also can adapt to the situation, so they can hijack this host inflammatory response. So that's the problem usually we are facing to control these viruses. So because they can change their properties according to this environment, and they can adapt more easily. So, and we heard about the cytokine storm. So it's related to this one. So once what will happen if the it's out of control, then more NF-kappa B or inflammatory genes will produce, and that leads to the multiple organ damages like kidney damage, lung damage, and liver damage that leads to this death of the patient. As I mentioned, so the pathogenesis depends on this inflammation. And once, in the normal conditions, once this pro-inflammatory cytokines or chemokines is released, then they will recruit more innate immune cells like macrophages. And then for the adaptive immunity, they will recruit this B cells and T cells. The biotechnology is actually having a huge role in the innate immune study. So not only in the research field, it's applied to the diagnostic and the therapeutics. So we are going through this next generation sequencing. So I am talking about the biotechnology, influence of biotechnology, how we are utilizing that one. So the recent one is the single cell RNA-seq. So from the single cell RNA-seq, we can look into what is happening in each cells. And there is a gene editing technologies, which like manipulating the proteins and other things in the cells and also in the animals. And also cell-based technologies like immunoassays and like in the ELISA and also in the antibodies for the Western blot. Then biotechnology is also involved in the next generation to generate the next generation vaccine adjuvants, and also the recent advanced or recent studies are going on with you familiar with this term nowadays. It's the training innate immune cells. So we will take the immune cells from the patients and then train them. That training means with a hit with the, these ligands, and so then they will they will convert into this activated them, and then reintroduce into the patients, and that will be useful to fight against the cancer and or also the SLEs like autoimmune diseases. So coming to the so we usually in our lab we are looking into the toll-like receptors and RIGI related to in the cytoplasm. So we found that this activation. So once this RNA or DNA is bind to the receptor, so it need to the activation. So usually the activation is through different modifications of the proteins. So we saw that they need this phosphorylation of tyrosine, which leads to the recruiting of the downstream proteins. And this tyrosine kinases or tyrosine kinases are important, and if we are blocking that one, then we can rescue the from the disease. Like I mentioned earlier, the interferon is good, and also it's bad when it's coming to the septic shocks. So if we are blocking the EGFR, then this downstream interferon beta production can be blocked, and then rescue these septic shocks in those disease conditions. Also, the same thing in STING. So the STING is then phosphorylated in the presence of EGFR. So this is another example for this septic shock. So we found in another situation, like this is DNA. so in that DNA, it's actually TLR9 is involved in these processes. And we found that actually the culprit is the TNF production. So if the TNF production is high, then you can see that it will cause the liver injury. So this is about the CPG ligands, and you can see that the liver is having the hemorrhage and apoptotic nuclei. But if we are blocking the EGFR, then we can see that it's a normal condition. But we also found that it's the recruitment of macrophage, or the number of macrophages are constant. You can see that it's a constant, but the death rate is high in the ligand treated. So so this is due to this leads to the so we went and looked into the macrophages, and we found that this TNF alpha is actually producing by these macrophages. So if we are removing the EGFR from the macrophage, then we can find that we found that we can rescue these mice. So ISTs are, as I mentioned earlier, ISTs are also antiviral. So if we are removing this gene, IST gene TDRD7, so we can see that more viruses is we can detect more viruses in that knockdown condition mice. So these are the lungs. So we can see that different. This is a normal lung, and this one is the diseased one. So there are so many infiltrated and damage is happening inside. So coming to the tumor, so I mentioned about how this the training of these immune cells. So usually we can activate this through the sting, like so we can activate the sting by introducing this DNA. So we'll take this macrophages out, or DC or macrophages out, and then they can be differentiated into the other immune cells, and then reintroducing into the patient, and then they can kill the tumors. So these cells will only target the tumors. And again, this is about the prostate cancer. So it's already reported that the prostate cancer patients are having the heart attack or thrombosis, heart-related diseases. But we don't know about this mechanism, how this is happening. So we found that this once in the tumor, they will release the small vehicles. It's called exosomes. And once this so these exosomes will contain these cytokines or chemokines or other inflammatory genes, and then it will travel through the bloodstream. So once it's detected the platelets, then they will bind to the platelets, and then will give the will start activates the proteins, and then it leads to the integrin activation. That integrin activations actually leads to the platelet aggregation, and then will form the thrombosis, and that leads to the disease. So if we are blocking with an antibody or gene manipulation, so we can rescue this process, like there is less thrombosis in that process. So So yeah, I mentioned about the recent Nobel Prize said about the regulatory T cells. So it's coming in related to the autoimmune disease. So in autoimmune diseases, what is happening? So usually our body is protected in such a way that these receptors will not detect our own DNA or RNA. But in some cases, what will happen if there is any gene mutation or other high level of cell death, then what will happen? After apoptosis of these cells, then they will release the DNA or RNA. So the DNAs will then detected by the this immune cells through TLR9 or TLR7. So then they release the cytokines. So that's how this disease is progressing. But in the regulatory T cells, they have found that so before producing the cytokines, there is these T cells, the regulatory T cells can identify these DNAs or particles like this is okay, this is the self our own DNA, so don't attack, or these are not intruders. So that's how the signaling is happening, and then so something is happening to the regulatory T cells, then the break is not there, and this disease will progress. Okay, so coming to the brain-eating amoeba. So we heard about this brain-eating amoeba, mainly with this amoeba nucleare. So what is happening in the pond or in the swimming pools? This so usually these amoebas are generally it's there in the outside in the soil and water, but normally it will not interfere with our system. So usually in the ponds, if this cyst, so this is a life cycle of the amoeba, cyst and then tropocyte. So these tropocytes, if they are entering into the body through the nose, then it will directly go to the olfactory nerves, and from there it will go to the olfactory bulb, and so then they can overcome the blood-brain barrier, and when they can enter into the brain. So once this amoeba enters into the brain, then it can go through two phases. Like one is phagocytosis. Phagocytosis means they will eat the cells, neighboring cells of this amoeba. Neighboring cells means the brain cells of our brain. So then another one is they can release the enzymes, so they will kill the so they will kill the cells, and then they can that will leads to this brain damage. So the same thing as I mentioned earlier, what is happening here is like we found it in COVID-19. The response is delayed, so that when the response is delayed, then it will recruit more immune cells, and then the immune cells will release this pro-inflammatory or antiviral, that will leads to the huge, that's called the cytokine storm. And that will leads to the severe brain swelling. And the disease is called, with this Naegleria, is called the PAM. And along with this amoeba, there are 3 more amoebas reported for the brain eating. So one is, and this sapenia is reported only 1 case, but that patient survived. But this actamoeba and balamuthia, so they are also reported in Kerala. So this actamoeba, these 3 amoebas actually entering into our body through the respiratory tract and the skin lesions. And the same thing, once it's so here what is happening, so it will go to the in our body and it will enter into the nerve, so neurons, and then it will go to the nerves and then enter into the brain. Once it reaches the brain, the same thing, the immune cells will recruit it and then severe brain damage. And that disease is called GA, granulomatous amoebic encephalitis. So as I mentioned earlier, these are rare incidents, but nowadays it's highly reported in all these around the world. So the main thing expecting is because of the climate change. And the another one is because of the co-infection. So the co-infection is once, like for example, if we are getting, we are went through this COVID or flu season. So once this is happening, our immune system is a little bit unbalanced. So these amoebas are some bacteria also there. That's why we are getting this pneumonia after following the viral infection. So there also it's a co-infection, like a virus will come and then after that the bacteria is coming. So this co-infection is also a hot topic recently. So we don't know. I'm speculating that maybe that's also a reason for this increase in the cases. So if we are looking at this immune responses, so you can see that these toll-like receptors are very high. So the inflammatory necrotic lesions are there. So maybe that's also. So coming to the future, so I mentioned that immunotherapy is one of this ones. So immunomodulatory stimulator modulators like cytokines, interferons, interleukin, or we can also target the immunomodulatory drugs. So that's how this biotechnology is also important to move this innate immune field, and also for this advantage of these patients. So we want to look into the balance between early protective innate response and the delayed chronic inflammatory responses. And about the coming to the training, so there I mentioned mentioned about the ligands. So some of the ligands are LPS, BCG vaccines, adenovirus. And even Hellman is a worm, but it can found that it can also give the trained immunity. And also, even though this LDL is not good, but that can be also used for the trained immunity, and also for the vaccine adjuvants. So the vaccine field is also moving forward. Like we found that it's mRNA vaccines are coming, but again, we need the adjuvants to activate, proactivate the immune cells and to get more efficiency of these vaccines. So vaccine adjuvants are also related to. Yes, so thank you for listening.
Thank you, sir. Thank you, sir, for that insightful talk. We now open it for questions. Participants, you can raise your hand. I think there are a couple of questions in the chat box. Dr. Srija is asking, will Naegleria fowleri infect animals? And if not, why?
Manoj can read that. Manoj, can you?
Manoj, sir?
Dr. Manoj, can you read that?
I am not familiar with this one, so where I can chat.
Yeah, she's asking if Naegleria fowleri infect animals.
I am not sure about because this is not my field. So yeah, I didn't see reports about that one. So I mainly concentrated on the clinical studies. So clinical studies are yeah, sorry, I cannot answer that one. Any other?
I think there are a lot of clinicians, doctors, participants.
Can you?
Yeah, one. And Rajdeep Mandal is asking if Naegleria fowleri can switch between amoeboid and flagellate form, and which form mainly attacks the brain.
Yeah, so I mentioned earlier, so these tropocytes actually enter into the brain, and in the patients, like after that, they found that the brain contains the cyst and the tropocytes. So the tropocytes is actually going into the brain and attack and release this phagocytosis and releasing the cell enzymes.
Thank you, Dr. Manoj, for the talk. I'm just wondering, like, in this brain eating, this amoebic infection, how this brain or why this brain blood-brain barrier do not pose any objection for the amoebic infection? Like, why it cannot prevent the amoebic infection?
Yeah, that's a good question. So that's not even amoebic, in the viral also the same thing is happening. So if you are looking at the COVID-19, the same case, so it's the same question raised, how this COVID virus can enter into the brain without so we know that a blood-brain barrier is important because that will stop this entering of this virus or any microorganism into the brain. But we also publish some work related to the virus. So I don't know more about this amoeba because it's still going on. But in the virus case, what is happening? The virus actually, so the interferons actually, so once it's entering into the nerve, so then they will, inside the nerve system itself, they can produce these interferons. So then the interferons will stop these viral propagations. But the virus can, as I mentioned, the virus can also manipulate the system so that what will happen, they will hide this interferon production. And once the interferon production is hid, then that will, then this virus can move forward and enter into the brain. So that's how we found that this one. So, and we using the gene manipulation, we shown that this interferon is important for this to restrict the entering of virus into this brain through the nerve. So I am not sure whether this amoeba is also doing the same thing. I didn't see any mechanical work related to this one. So once they are, they can go through this, they somehow restricted this one and enter into. So that's the same thing. So the cytokine storm in related to the COVID-19, the same thing. It's published. So this virus can hide or suppress this innate immune responses, so that the body will not detect it initially. But once they enter into the brain and then they propagate in there, then this more immune cells will come there and then start releasing a huge amount of cytokine, and that leads to this cytokine storm. So that's how this brain, the same thing is that. So it's two effects. One is with this amoeba is phagocyting and then cell disease, and also the host immune cells are also coming there and then releasing this cytokines, and both of them will damage the brain. Is it clear or?
Yeah, thank you, Doctor.
Well, one more question in the chat box. Person it's online, they can interact.
Can you, because I don't know where is the chat box in this.
Okay.
Fine.
When you train the regulatory T cell at the in vitro condition, how do we train them to differentiate the receptors of indigenous gut consortia, since they are dynamic in nature? Do we go for metagenomics of the specific patient undergoing treatment? That's the question being asked, relating to the gut bacteria.
Yeah. Yeah, so the actually I am not working in the T cells. I am working in the innate immune cell. But what I understand from the literature, so these yeah, so I can correlate with the macrophage. So the macrophages, there are M1, M2 macrophage lineages. So then if we are treating with the ligands, and then we can convert them into the active macrophages, and then we'll put it back. I am not expert in the T cells, so sorry, I cannot answer that part of that one.
Okay, one more question. Why the innate immune response to rabies virus this week?
Because recently we heard that, children are dying even after vaccination.
Yeah, so there is a, so that's the problem with that. That's why I initially, when I introduced the adaptive immunity, that time I mentioned that this that adaptive immunity is even though in the childhood, some of the vaccine, I mentioned, stress that some of the vaccines are useful for the long-term protection. So because the vaccines are, we can categorized into two. The one vaccine is it can protect the long life, but some of them are there. It's a weak antibody production and then a weak response, so that it can protect only for a few months or a year. The same thing if we are looking at the flu vaccines, we need to take it regularly, like yearly. Now the COVID vaccine came, when the COVID vaccine came, we thought it can last long, but we found that the antibody production is high in the first few months, then it's going down. So it's the same thing. So in the rabies, what is happening is I looked into this one because it's a debate is going on. So what I understand is it's not because of the so these patients are actually getting the vaccine after the bite, and we know that the part of the bite, if it's near to the head, then it's very risky for because it's very easy for the virus to enter into the brain than if it's getting like say the leg or the downside of the body part. So the because the virus will take time to get into this brain. In the meantime, so in that duration, the vaccine can act. But if we are, if the bite is near to the brain, then it's very difficult. It's the virus is going very fast, faster than the recruitment of these memory cells, and then getting this infection protection. And most of the cases reported in Kerala, after death is after getting vaccine. I think it's because they are getting vaccine after the bite. So that's main reason for that one. Yeah, and it's the same thing. The virus, there are studies going on whether the virus is mutated in an Indian situation. Like if the virus is mutated, then the vaccine, we need to change the vaccine, like in the flu and even in the COVID. So updated COVID vaccines are coming because the COVID virus is changing its genome, like mutations are happening in the ACE, not ACE, the spike proteins. So it's the mutation is happening there. So we are always targeting the spike protein. So if the spike protein is modified, then whatever we target, the antibody will not detect this new mutated viruses. So yeah, so in Indian situation, I think scientists are looking at whether the mutation is also happening in that case.
In this case, by one.
Is there any other
Phase.
question? Pardon?
That person can unmute and ask.
Sorry.
Are you able to see that?
No, the problem
It's
is
about-
I
Yeah, it's about
don't know.
macrophage.
So where I can find the chat and how I can get.
How to, okay. In the
Maybe I
right
need-
corner, there's
That icon, sir.
I can, yeah.
Yeah, got it. Yeah, so regarding this identification of M1, M2, yeah, these are CDs are actually we are looking at the CDs for this changing in macrophages.
Maybe you can share the presentation, then you can see that.
Yeah. So I looked back. So most of the questions are answered. Yeah. So the last question is about M1, M2. Yeah. So we usually look into this CD markers for the macrophage modifications. Yeah. So any other
So,
question? I
can you
missed?
tell us, for the student community, can you tell us more on about your research journey to innate immunity? The advice that you give to the student community who are interested in this particular area?
You mean next generation sequencing or?
No,
So what's your suggestions, for the new generation students,
Oh, okay.
from your experience? Maybe you can give some suggestion, ideas, direction for their career and all.
Okay. Yeah. So this if you are interested in innate immunity or this one, so yeah, it's a moving like advancing field, and because of this, mainly because of this COVID-19. After COVID-19, then it's a huge jump in this research, because we are now aware that there are so many other viruses or microorganisms out there, and because of this, actually because of this change in climate, more and also because of the this our behavior with the environment. So we are getting more viruses or these pathogens than before. But our so each virus or each organism is different, and we need more study for this one, how we can, because we know how to tackle the virus, but when the COVID-19 came, then we were in a dark, like even though this is coming from the other SARS and SARS and MERS, and we have the coronavirus already out there, and some of them are like we are calling as flu, like cold virus, but where the same family, the same thing, the COVID is also in the same family, but when it came, then we stuck because we don't know more about that one. But we tackled that COVID-19 because of this previous knowledge and the new technology. And that's why I mentioned earlier that biotechnology is important for this moving forward. So we need to club like interdisciplinary manner. So we need to club all these together to move forward. So wherever you are, you need to be means wherever you, like if you are in biotechnology or in basic research or in a clinical research, so wherever you are, so you need to look into your field, grasp what is going on, and then move forward with a challenge, and then move forward. So that if you are looking at this Nobel laureates recently got the So most of them are went through this pathway, like they got some hints, and then that's why in the Nobel Prize for Physiology or Medicine, there's a regulatory T cells. So they found in the mice that some of them are dying, or they are getting injured. So then they went into that why they are because it's only in a few percentage of population. They saw in their animal population they saw this one, but they went to tackle that problem. And so that's why you need to observe what is so just not doing things, but you need to observe what is going on. And if you can tackle that one, then you can get a new area to move forward. Yeah, of course, you need financial support from the funding agencies and other, but the main thing is you need to show that, okay, you can do this thing, and then do. So be creative, and then yeah, normally hard work, and then think about your ideas and try to get into that. Yeah, that's.
Thank you so much, sir, for that highly insightful and engaging presentation as well as your talk beautifully illustrated how the innate immune system forms the foundation of our body's defense and continues to thank you so much for joining us today. I now invite Arjun Prasad, the scientist of KSCST, to deliver.
Thank you, Dr. Sharika. Good afternoon to one and all. On behalf of this organizing team behind this webinar, I express my sincere gratitude to Dr. Manoj for joining us today and enlightening us with your presentation. It was, as Dr. Sharika rightly pointed out, it was very informative. Started right from the story started from innate systems, how it the different mechanisms involved. You touched upon the application, how the field of biotechnology can contribute to the innate immunity studies. Then you connected well to the recent amoebic infections, how it happens, or the different mechanisms involved. Thank you very much for your talk and for joining us today. On behalf of KCST and our entire team, I express sincere gratitude to you. Thank you all participants for joining us today. I hope you had a great time, and it was worth listening to one of the researchers in this field. Hope you were benefited. And Kerala Biotechnology Commission will be conducting similar webinars in the coming months, and we will keep you informed. Thank you. Thank you for joining.
Yeah, at least some of you can switch on your video, take a photograph. So thank you once again. Kerala Biotechnology Commission. Big thanks to the speaker and all those who have joined today. And we look forward to our continued support and engagement in our future programs. The next month program also. Thank you all and have a pleasant evening.
Thank you.
Thank you.
Samedra. സമയം എത്രയാ മനോജ് അവിടെ?
6:35.
നേരത്തെയാണോ അതോ താമസിച്ചത്?
ഏതാ?
നേരത്തെയാണ് താമസിച്ചാണ് എഴുന്നേറ്റത്? നേരത്തെ എഴുന്നേറ്റ്
കുറച്ച് നേരത്തെ എഴുന്നേക്കേണ്ടി വന്നു. 1 hour.
മനോജ് നെക്സ്റ്റ് ടൈം വരുമ്പോ കൗൺസിൽ പറഞ്ഞു KCST പറഞ്ഞു കേട്ടോ.
ട്രൈ ചെയ്യാം.